Yes. Sorry, Georgia. Welcome, everyone. Thanks for joining us. I'm really pleased to introduce our data science speaker for this week, who is Louis Stan from Indiana University, where he's an associate professor in the math department. And he's also currently a research associate in the Department of Organismal and Evolutionary Biology at Harvard. And today he'll be talking to us about spatial stochastic modeling for population dynamics.
Thank you for the introduction, Jasmine, and thank you so much for the invitation. It's a great pleasure to be with you here this afternoon. So my talk will be on spatial stochastic modeling for population dynamics. I'm going to talk about some recent work on analyzing spatial temporal data. A lot of the work are done in collaboration with my student Johnny Yang and also collaborator John Law and also John Yin, Wisconsin Madison. So let me first, it's not the next slide. So, oh, thanks. Right, so this is the outline of the talk. I will first give some quick motivation and background on population dynamics, and then talk about a new and robust model for population dynamics. And then I'll give a theoretical applications for an idealized model, and then some data application. So this is a data science seminar. And then some future directions. Some quick motivation. I'll show two pictures, essentially. This is real spatial temporal data taken from my collaborator John Yin's lab at Wisconsin Institute for Discovery. So maybe let's focus on the first row. So there are 16 images here. Let's focus on the first row, the 4 images there. These are 4 images taken at 4 different time points in a virus infection experiment in the lab. So 7 hours post-infection, 13 hours post-infection, 19 hours, 25 hours. Let's focus on the top left little square. So this is an image of a petri dish with about millions of susceptible cells. And then the middle cell, or the cell at the center, was infected by a certain wild-type virus, the so-called VSV virus. And then the virus will release its genome and then turn the cell into a factory to produce virus progeny. Now that middle cell is co-infecting over time. Virus will butt out of the cell and then infect neighboring cells. And so over time, the population of infected cells will grow in size. So these are the first 4 images. Now, the second row and the third row and the fourth row are essentially the same experiment, but with different initial condition. So let's say the second row, now the cell at the center is co-infected by one of its mutants. So this shorter green bullet, one of its mutant, which has a defection in its genome. Now, both the virus, the wild-type virus, and its mutant will produce progeny, and then we see different spatial patterns. Oh, the third row and the fourth row have a higher initial dose of the mutant virus, the green shorter bullet virus. The details are not important here for now. And by the way, the spatial scales here is 1/2 centimeter by 1/2 centimeter, about. Well, important question I should ask is, how can one devise or find some kind of mutant virus particle, how they co-infect the cells, how they give rise to different spatial patterns? Can some kind of mutant stop the virus, stop the wild-type virus, for example? And so Yin's lab has the expertise to genetically modify the virus particles and then systematically initiate and observe the virus infection. The second spatial temporal data is this growing bacterial colony. So this is a remarkable experiment by Holdershek, Nelson, Herson, Ramanathan on E. coli cells. So this is an agar dish now, full of nutrition. And 2 strains of these E. coli cells grow on this agar dish. And then even though initially it's well mixed, over time we see different spatial patterns. Of course, there are other population dynamics. We can talk about population of living organisms or plant propagation over seed dispersals over many, many years. But some general motivations are what govern the population dynamics, whatever population we're talking about. For more precisely, what is a change in population densities of different genotypes or different types over space and time? And when a mutation arises, will it go to extinction or fixation? How will it interact with different types? What determine a macroscopic or observed spatial patterns, such as the propagating speed and different spatial patterns? But whatever question we're asking, we need to understand the mechanism behind the model in order to make predictions, in order to explain spatial temporal data. For example, for cancer infection, cancer growth, we need to understand the mechanism behind in order to predict what will happen if we apply new drugs. Right. So here are two very big general mechanistic modeling approaches. The first is the individual-based or agent-based approach, where we keep track of every individual. So let's say every virus particles at every locations. But there's another approach, more macroscopic point of view. We don't care about every individuals, but we care about the overall density or overall abundance of the different individuals, and they have different pros and cons. Right? So the individual base is easy to specify, but often computationally and analytically intractable. The macroscopic approach might be more amenable to calculus or analysis, but if you write down an equation, we need to understand or make sure the equation is correct and what those parameters means in terms of the microscopic dynamics. And of course, there are many other modeling approaches like hybrid approach, multi-scale approach. But the key is to understand the connection among different models. And this is part of the theoretical application of this talk. So let me talk about the macroscopic approach. By the way, feel free to interrupt if you have any questions or anything not clear. So what are the sort of canonical macroscopic modeling approach for population dynamics? We describe a population density at time t and location x. Roughly speaking, it depends on whether we take into account of randomness or spatial interactions. So you can imagine we can fill in these four logs, and Yt, let's say it is the population density at time t, and then Ut, comma x is the population density of some type, right, at time t and location x. Let's look at well mixed. Well mixed means it's spatially homogeneous, and we don't have different, we don't have variation across space. We just care about the number over time, right? And well, this is, by the way, real time series data from the same experiment. We take the middle cell or one of the cell and then look at the virus abundance measured by gene expression levels over time in a 10-minute interval. And this is just two examples of the equation. OD is ordinary differential equations, or SD is the stochastic differential equation. So these are, so this is probably one of the first model, logistic growth model for population dynamics, right? There's a carrying capacity, here just one after normalization, and beta is a growth rate. And the SDE here is the classical right-Fisher diffusion, which is used a lot in population genetics. And there's a new parameter, the gamma there, an extra parameter here, and an extra term here that capture what is not captured in a deterministic model. It can model randomness. It can also model ignorance, right? So it's a noise term, and this BT is the Brownian motion. And what about spatial models? I'm going to show just a quick example, right? And analogously, by the way, this highlighted term here, this beta U1 minus U is the same term that appear, but this extra noise term here, there's this extra spatial motion here. And in this talk, we focus on more continuum space. So space is more in a continuum. So this Laplacian is the second partial derivative with respect to space, model spatial diffusion in continuum space. And this alpha is an extra parameter, the diffusion constant, that tunes how fast the individuals are dispersing or moving around. And this is a PDE model, partial differential equation. And the last one, stochastic partial differential equation, is you can see it has both a spatial motion term and also a noise term here. But the noise here also has a space here. And this one is the space-time white noise, which is essentially a distribution value Gaussian process uncorrelated in space and time. And this equation arises quite naturally as sort of a spatial version of the stochastic of the Wright-Fisher diffusion model. This equation, there's still some open problem for just this equation or a similar equation. So stochastic PD is much less studied or applied. And I will talk more about that equation soon, but let's spend the next few slides on the PD and the stochastic PD we just saw, okay, to conclude this background section. So with the Laplacian term, this is the heat equation. We know how to solve it. We can, this PTXY is the Gaussian density function, the transition density of Brownian motion with the variance 2 alpha. And this is the Fisher KP, as you mentioned, this is called Fisher KPP equation. And it was introduced a long time ago. And the most important prediction of this PDE is the existence of a traveling wavefront. And it has been proved in different ways. Maury Bramson, who was here, have some very delicate work, important work on getting asymptotic wave speed by using branching Brownian motion and get lower order asymptotics. So the asymptotic wave speed was shown to be 2 times square root alpha beta on the same alpha beta coefficients up there. So PDEs are powerful, but it is not adequate to explain the spatial temporal data we saw, because the movie we saw in experiment are never the same, even with the same sort of initial condition and all the same setting. So we need to take into account for randomness. That's where stochastic PD kicks in, where we have an extra term there that captures the noise. And this example is a stochastic FKPP equation we saw in the lower right corner of that 2x2 block. So in general, analyzing stochastic PD are challenging. And as a matter of fact, even just for this equation, and in fact for almost all reaction diffusion equation driven by space-time white noise, there's no solution theory for dimension 2 or above. And so my work, part of my work is to understand these stochastic equations that arise naturally. So I'm going to talk about a new class of stochastic PD that come from explaining a class of population dynamics motivated from virus infection experiment, and then a theoretical applications and a data application. So this class of stochastic PD are a stochastic PD that with space given by any metric graph. So what is metric graph? Metric graph are just a graph whose edges are intervals. For example, the real line is a metric graph with one edge and no vertex. The circle is also a metric graph with one edge and no vertex and is compact. The interval is also a metric graph with one edge and two vertices at its endpoints. Make sense? And there are many others, right? And instead of using a petri dish, Yin and his collaborator at Wisconsin, they manufacture and use this microchannel as the container for cells. So they put the cells in these microchannels to model different kind of tissue or system like an artery, brain artery or blood vessel or graft, and then systematically initiate and observe virus infection. And so we want to understand how this spatial structure affect dynamics. And we care about the so-called defective interim particle. This is the main sort of object of study in all this kind of experiment, which is the mutant I mentioned. It is the mutant virus that arise naturally and it has defection in its genome. So the genome length is shorter and it has defection in that it is not infected by itself. So if a defective virus particle, the defective interfering particle, infect a cell, it cannot produce progeny. But if it co-infect a cell with a wild type, then it can produce progeny at the expense of the wild type virus. And then our modeling framework is to look at a class of reaction diffusion equation with noise term, or as a class of stochastic PD, as I mentioned, on metric graphs. So we mostly look at the density of the wild type virus particle and the density of the defective interfering particle. So this is the framework. So let me spend a slide to talk about what is this equation. Let's just focus on one entity, this U. There's a spatial motion term, this L of U, might be a Laplacian. And this drift term B, modeled by chemical reaction networks, maybe a usually polynomial term by a mass action kinetics. Sigma is the noise term coming from that, like a Langevin approximation, but with spatial component. And then for simplicity, we assume flow in equals flow out. So the boundary condition at the vertex is just a so-called Kirchhoff boundary condition or no flux boundary condition or gluing boundary condition, there are different names. And mathematically, one way to make sense of a solution is to view this equation as a short form of this integral equation. Just like for stochastic ODE, Brownian motion is not differentiable. And then we look at the SD with respect to Brownian motion, we'll view it as a short form of an integral equation and then make sense of that equation by making sense of stochastic integration. That's where stochastic calculus kicks in. And same here, we use this kind of a Duhamel formula in PD or variation of constant, right, formulation. And then if we can make sense of this space-time integral with respect to space-time white noise, then we can make sense of this equation. Now, this PDXY is like the Gaussian transition density we saw before in Heat's equation. But for graph, it means the transition density of the Brownian motion on the graph. So this is something a little bit different, but you know, one can still handle it. But so the analysis of this equation rely heavily on what we know about the underlying Brownian motion on the metric graph. And here you can see why 2-dimension can be a trouble, because in 2-dimension or above, the transition density is singular, right? At time equals 0, very singular. And then this term will have infinite variance, and it does not make sense. But for metric graph, it's okay, because most metric graph is okay, because it's essentially 1 dimension. For some fractal, it's also okay too. So this is the equation, and why do we look at this? First of all, it arises naturally, and this is most important. These models are sort of a generate. It arises from discrete models, and then it bypasses the trouble for high dimension, and so enable us to analyze the effect of space. So let me talk about any questions for now? Perfect.
So, sorry.
yeah, yes.
Did yours say your collaborator did this experiment on like graphs besides just like the 4 lines, like more complicated like brain tissue or something, or?
See, so in practice, they can put in this little
The micro-
channel.
Okay.
In whatever graph they want.
Okay,
So the comb graph, right, or the caterpillar tree is one, and the regular tree is also one way that we're looking at. Theoretically, they are more complicated one, but
so there were 2 examples of what they actually did, were the column graph and the tree.
yeah, the regular
Okay,
tree, yes. Yeah, but yeah, you're right. They're more complicated one, but they are still ongoing work.
okay, thanks.
But hopefully, some of the theoretical result that predicts maybe traveling wave speed, can inspire future experiments. So, yeah, thanks for the question. So how does PD and stochastic PD arise from microscopic laws? Let's focus on a simple example here in this section. So I will talk about, again, this stochastic FKP equation and explain how it arises from microscopic model. And I will use also one example called the bias Wai-Ton model, how it arises from it, maybe also its variance. So the bias Wai-Ton model is also called a spatial Moran process. And it was probably first introduced to model the interaction of healthy cells and cancer cells. And it was used to study cancer progression and initiation. And it also was used to model opinion propagation. And it was also analyzed in the context of interacting particle systems. So let me talk about what is a bias Wai-Ton model, right? So imagine let's use us as an example, right? Let's say all of us is a voter, right? And each of us has one of the 2 opinions, let's say apple or orange, but not both. And then our opinion will change over time. So maybe now I'm an apple person, but after some time, James and I are neighbor, and then you may spread your opinion to me and say orange is better, then I would change my opinion, right? And then let's say, right, the Chang Yu is not my immediate neighbor, and Anab is my neighbor, so maybe my opinion is spread to Anab spread to Chang Yu over time. So to specify this model, I need to tell you who are neighbors, right? And the second, how often are we spreading opinions? Right, these 2 things. So here are the sort of a spatial structure. So there's a 1-dimensional integer lattice with spacing 1/L, or L to the power -1. And then each location has M individuals. Let's say there's a 4 individual, M is 4 in this picture. And then each individual has 8 neighbors, 4 to the left, 4 to the right, or M to the left, M to the right. Now, these 2 colors you can imagine are 2 opinions, maybe apple and orange, right? Type 1 and type 0, let's say. So type 1 and type 0. Type 0 is this open dot, maybe orange. Well, it reproduces at a rate proportional to L squared, where 1/L is a lattice spacing. And then immediately after reproduction, then the offspring or the opinion will spread to one of the neighbors chosen at random. Now, type 1, which might be this solid, maybe apple, has a faster rate of spreading its opinion. So this beta is a bias. That's why it's called bias Wai-Ton model. And this little bias will cause the dynamic to be different. Is the model clear? So let's take a look at a simulation for this bias Wai-Ton model. With L equals 10, that means 0.1 is a lattice spacing. M is 1,000, so there are 1,000 individuals at each location. And then the rate is 200 and 204. This. So this is a simulation of this bias Wai-Ton model. 1 means all the 1,000 are of type 1. 0 is all the 1,000 of type 0. 0.4 means 400 of type 0. Sorry, 400 of type 1 and 600 of type 0. Yes,
What's happening at the boundary? Sorry if I missed it.
boundary,
You have 1,000 sites.
yes.
And at the boundaries of the top and the bottom.
It's just, yes, so if it is at the boundary, let's say at this location, if all the 1,000 individuals are type 1, then we put a circle there. If at this location, let's say at this location, there are 200 individual here.
Sorry, I mean like for the individuals at the boundary, do they just have less neighbors? Or is it
Okay,
periodic?
good point. So the model we're looking at is the infinite lattice, but in the simulation, you're right.
Oh, okay.
You're right, in a simulation, right, good point. In a simulation, here is already type 1, so it doesn't matter. Because it's fixed. Yeah, good point. Yeah, the theoretical model is infinite lattice. The simulation has a boundary, but it doesn't matter because of the initial condition.
Got it.
And the red curve there is the FKPP, which we know the speed is 2 times square root alpha beta. And we see that it's not quite a correct prediction. And this is kind of annoying because this simulation takes a few minutes to run, with 1,000. But if you have 1,000,000 of individual, then it will take much more time to simulate for one fixed parameter. And sometimes we don't know the parameters. Oops. Sorry. Right. So there was this theorem with Rick Durant quantifies the relation between the bias photo model and the FKPP equation, or the stochastic FKPP equation. So we show that if the ratio alpha m converts to something called gamma, then that gamma will pop up as the parameter in the macroscopic stochastic PD. So this is the connection. And so this connect the model at 2 different scales, and it enables efficient simulation because we can simulate the stochastic PD as an approximation of the bias for the model without caring about how big the number of individuals are. And this also gives us a notion of a local effective population size in this spatial setting, which is proportional to the number of particles at each site at a unit interval. So 1 unit interval has L many sites, L many site, and each site has M individuals. So just to summarize, we have a bias photo model, we connect to a macroscopic equation and connect the parameters, understand it correctly through in terms of the individual base model. And from this, we can look at theoretical result we know about this equation. And it was predicted by Brunet Derrida and later proved by Carl Miller, Mitnick, and Gustav that for small noise, when gamma is small enough, the speed is little slower. This is the 2 times square root alpha beta of the FKPP equation. And this is the correction, the first order correction by this result. And numerically, they are pretty correct. We can see in a simulation before, the red curve is, where is it? Is ahead, right? The bias for the model is a little slower than the red curve. You remember, I should point out that. And this is the slowdown.
So we,
Yes.
what kind of convergence, like what is the metric space? Is it finite dimensional convergence or?
Good point, good point. These are all for any arbitrary time interval, when M and L are big enough, these two are close. So this is the correct rigorous statement.
In the supremum norm.
Okay, good point. Yes, supremum over space. Yeah, we're looking at the supremum now, the C space, continuous space with supremum. So this function are all bounded between 0 and 1. And we're looking at, well, okay. The norm we're looking at is the local uniform convergence.
Is it also for one dimension or I didn't know what the dimension is. 'Cause I know you said
No,
this
you-
equation doesn't have.
This is one
solution.
dimension. This is all one dimension.
Yeah.
Yeah, so I'm using this as an example. That's a good point. Yeah, in two dimension, we don't know what this equation means
Yeah,
rigorously, but the important point is that, well, we can modify the bias photo model, right? Right now, we look at left M, right M as a neighbor. But you might say, why don't we look at the left 4 columns and the right 4 columns as neighbors, right? We can do this, we can modify it, right? Or we can look at the same column as neighbor, right? So we can modify this model in many ways. But the important thing is that as long as the modification is not too drastic, you still get the same functional form of the equation. That's underlying the robustness of this equation. Yes. There are other results that along this line. Right. Yes, is that so this class of equation for mass action kinetics arise as robust scaling limits. And in recent work, we have we study the FKPP equation on a class of random regular trees and obtain the traveling wave in terms of the degree of the tree. And we also got a critical birth rate. So the contrast is this. Remember, the FKPP equation has traveling wave for any alpha beta. Whenever beta is positive and alpha is positive, we get a wave. This is no longer true for if we spread the wave on a tree. It can destroy the wave if the birth rate is not big enough. Because you can imagine if you have a tree have a lot of branching, it will spread out the wave, right? It will spread out a lot. So that's a critical birth rate that the birth rate has to be bigger than that. Otherwise, that will not be a traveling wave. And then with Johnny, Lin, and John Yin, we also look at extinction probability. And then recently with Oliver Tuff, we study the time to extinction and the so-called quasi-stationary behavior of the stochastic FKPP. So, yeah, maybe so the second part of the talk, well, okay, the last of this section is the data application. So this more recent work on applying the data we saw before about this virus infection on petri dish. So actually, this section highlights sort of a struggle, I guess, in a lot of interdisciplinary research where you can only prove theorem for idealized model, and it's hard to prove theorem for realistic model that compare with data. This is also the case here. But we have a recent work published in PLOS One to compare with experimental data. And now we have a reaction diffusion equation that describes the virus and diffractive interfering particle dynamics using 8 equations. So it's the same kind of virus particle and it's a diffractive interfering particles, the VSV virus, the same kind of cell line, the baby hamster kidney cells. And we look at basically the pathway, how virus and dips are produced. And the virus density and the dips density solve the reaction diffusion equations. Empirically, we know the diffusion for the dips is a little faster because it's smaller. And there's virus production and also degradation. And then there are 6 other equations that describe the density of 6 cell types, including the uninfected cells, cells infected by virus only, cell infected by dips only, cell infected by co-infected by virus and dips, and the cells that are mature enough to produce virus and dips. So there are 8 equations that here that I did not show, but you can imagine it will be a reaction diffusion equation taking into account all these reactions, right? This is the PD, but what about the stochastic model? We have 2 different stochastic models that have the same PD in the limit. So we look at 2 different scenarios biologically. It means that scenario one is when the cell, the virus and dips are produced by cell bursting. So after about 25 hours, the cell will die, it bursts, virus and dips will release, and then infect all the cells nearby. And scenario two is before the cell dies, actually some of the more selective virus particle will bud out of the cell and then go to infect neighboring cells too. So we look at these two scenarios. And we develop a hybrid simulation scheme to simulate this model. This 8 equations with the randomness. So basically in the simulation, we discretize the spatial domain into compartments, little squares. And then within each compartment, we assume the compartment is small enough so that reaction within the compartment is well mixed. But then there's a spatial movement between compartments. So compartments that have a small number, we will use a continuous time. We will simulate the interaction or the chemical reaction by a continuous time Markov chain, using Gillespie algorithm. But for region with large number, we simulate using a PD. We couple them suitably using so-called pseudo compartment method. But this is a schematic figure to mention. For each reaction, we look at compartment that have small number and then use the Gillespie algorithm. For big, for compartments with a large number of molecules for all the reactants, we use a deterministic model. And then this is the moving interface. It will move over time. And then with that simulation scheme, we can efficiently simulate the spatial stochastic model. And this is still the experiment we saw the first row, really. Scenario one, the cell bursting. Scenario two, continuous production. And then when we increase the number of dips, defective inner particle, we see a more patchy pattern. And so we look at two macroscopic features between the experimental data and the simulation data. This is the growth rate. So how fast it is, the patch is growing. And over time, remarkably, the patch is growing pretty linearly for both experiment and the simulation. And then that slope is the growth rate, what we call growth rate. We also look at patchy pattern, so how patchy it is, by using a summary statistic called the Q statistics. Basically, if you give me an image, I can use this statistic to tell you how patchy that image is. And using so-called Q statistic, basically is measuring what's the variance. Well, this statistic depending on the choice of so-called strata or sectors. In this case, because it's rotationally symmetric, we divide the image into like 30 pieces. It's like a pizza, 30 pieces. And then variance among those 30 pieces, among sectors, would be roughly the Q statistics. And then we compare the Q statistics in the experiment and also in the simulation. We see that scenario one, the cell bursting is more patchy because probably it has more noise. I should also mention that scenario one, it also has a smaller growth rate, probably also because it's more noise. Noise will slow down away. So in summary, we developed a spatial stochastic model for virus and dip dynamics, and also 6 cell types, right? 8 equations. And then we develop an efficient hybrid method to simulate this stochastic spatial dynamics. And it qualitatively captured 2 macroscopic features, which are the spreading rates and also the patchy patterns. Yeah, there are many future work, as you can see. The way that we deal with image data, I think there might be a better way, better image processing method, and definitely analysis of a stochastic PD itself. Yeah, we are still looking at different stochastic PD to analyze, understand extinction versus coexistence, the effect of different spatial structures. And we also want to look at more higher resolution spatial temporal data for the VSV virus, and also test the model using other model virus. The particular one we want to look at is the mouse coronavirus, which is a model coronavirus. This had also a short genome, but 3 times larger than the VSV virus, but still a lot is understood. And most recently, we started to look at single cell RNA sequence data for the single strand RNA virus, the VSV, and model genome evolution with insertion and deletion. Let me show one, maybe last slide of the motivation of this, looking at genome evolution with indels, is that deletion is observed in general for RNA virus. And so this is real data from we studied here, Wang et al. 2022 with real COVID swab data from a single patient. And every dot here is a virus genome with deletion. So let's say this dot has the starting position of the deletion is around 10,000 and the ending is about 20,000. So there's a large deletion around here. And then you see a lot of dots are near the diagonal. That means a lot of deletions are very short, start and close are similar, right? But there are some sequence data have a large deletion. This one is highlighted because this particular deletion seems to be found in different patients. Yeah, so I thank my collaborator mainly, John Yin in Wisconsin, in his system biology lab, and also my student Johnny Yang and Lin Wang. Lin might be on Zoom now, and Johnny might be too. And there's some mathematical work I mentioned also in my slides before. Yeah, maybe I'll stop here and take questions.
Additional questions?
Yeah, thanks for the talk. So I wanted to ask a question on the theorems you had earlier. So you had the convergence proof that you would proven where if you scale the parameters, right, you converge to the Fisher KPP with noise, right?
Yes, yes.
Then what I don't quite understand is the next slide where the wave speed is smaller.
Mhm.
Is it that you're trying to understand how the noise affects the wave speed, or?
Yes, or let's say you want to explain the simulation, right? In the simulation, we saw
But is
this.
it just that if there was no noise, then you know it's root alpha beta times 2,
Yes,
right?
yes.
But when you add the noise, that's when you get a slower wave speed, you're saying?
That's one way to get a slower wave speed. It is proven. Yeah, so it is it is in general known that for pooled wave. So when there's this different kind of wave, and a pooled wave is when the wave formation is formed by the leading edge. And the leading edge, if there's some growth that sort of pull up the wave, which is the case for stochastic FKBP. And it is conjecture in physics, not just for FKBP, but other pooled wave as well, that the noise at the leading edge will slow down the wave. And they have asymptotic for how it slow down the waves. And here, you're right, this is still an approximation, right? And then theoretically, it was proven by Miller, Minnick, and Costel that for this and this kind of equation, the slowdown of order log gamma squared. That gamma is 0.04. So this is the result for this.
Okay.
You're right. And then we also observe, independently in a simulation of bias photo model, there's also a slowdown. And then so we can empirically compare the speed, right? Measure the speed in the simulation and compare with this, and they're very close.
Okay, thanks. If you did it like
Yeah.
in 2D or something, like say you had a, start with like a circular group of voters or
Uh-huh.
something, and then it's gonna spread out like
Yeah.
this. Do you see the same kind of wave speed, or does the wave speed change over time in that setting too?
For the PD, I didn't show the simulation for the PD, but the PD for this is very symmetric.
Yeah.
And it does not capture the patchy pattern. And your question is, did we look at a noisy version of the
Is
PD?
there a theorem in that case, or there's not because you can't talk about 2D?
Good point, yeah, we don't have a theorem for that. Yes, yes. For the noisy PD or stochastic PD. Because
Even
we actually-
in that case where you could reduce it to one-dimensional in some radial
Oh,
trick or
yes.
something.
Yes, right, yes, we can look at the radio version and then study that equation. Right. Yeah, that's a good question. Yeah, maybe the 2D version is what we should prove a theorem.
I have a question. In the hybrid simulation part, as you said, is a moving threshold. I was wondering how to numerically decide this threshold. And,
Oh, that's a very good question. So far, we're doing some, in the paper, plus one paper, we're doing pretty ad hoc, basically in the experiment. If in experimental data, these movies, they have an intensity. These are intensity by the gene expression. So there's a experimentally decide the threshold. If it's too small, then we cannot detect. And so we use that, and then I think we multiply by 5 or something. So we ad hoc. So short answer is ad hoc. But I think there are ways to make it better, to have a more theoretical decision of what is a threshold, how to decide when it's small, when it's large. And one way might be to look at the extinction probability, because if the number is too small, it has a very high probability to go extinction first. But if it's big enough, let's say if there's internal equilibrium, then it will have a very small probability of going extinction. So maybe there's a theoretical threshold, and a result one can analyze. Yes.
I guess there's also another noise to consider in the sense that these different cell or compartments could have essentially different underlying parameters, right? They're heterogeneous in that way. Is it clear how to disentangle maybe that source of noise with the noise that you're considering here? Have you thought about?
Good question. So no, we did not. It's a good question. Even for different reactions, it was a different notion of big and small.
Yeah.
And
Yeah.
we did not look into that. It's a very good question. Yes. So it's ad hoc in this paper. And I don't know if anyone see, whenever this hybrid simulation occur, I think hybrid simulation is seem to be getting more popular. If you've seen any rigorous result on how to decide, what's the interface between the hybrid scheme, please let me know.
I'll just like follow up on that.
Yes.
I think Dina was asking about like how do you decide where the boundary is? But
Mhm.
like, I was just curious how you actually took care of the interaction at the boundary.
For the bipolar model, we just count the numbers.
But
Because
then to
always-
go, and then with the PD model though, like at that interaction, how did you?
in simulations or in?
In the simulate, like in this, so you had this hybrid,
This
hybrid,
one?
yeah,
Okay.
like how do you,
Oh, okay.
do the handshaking between the,
This transition. right?
Yeah.
Okay, this is the interesting part, actually. So this is called pseudo compartment. And so there is a transition between numbers here. Number of particle, number of reactants, right, number of certain particle to density. And then here, the PD part, we still have the density. But under suitable scaling, we can integrate the density to call it the number. Now it's an integer. Now, as integer, we can do the Gillespie algorithm to increase, decrease with certain rate, right? And then to transit back to the PD, we integrate it again. We normalize it again. So we divide by a certain normalization and call it the density. Does that make sense? So there's two, there are two,
Yeah.
you're right.
Do
But-
you have to add any noise or something? Because there's, and I'm just curious because I've seen this kind of thing before.
Mhm.
Where you're like tracking a wave and tracking a shock, and inside the shock region, there's a particle system, and then outside there's a PD.
Ah,
And I've seen like artifacts right at the boundaries that have to do with like, I don't really know, but some sort of numerical.
oh, good point. Yes, yes, you're right. The transition has to be careful, otherwise we create a systematic bias, which we don't want. But it turns out that for the number of, so when you switch from PD to Gillespie, after we integrate with respect to the area of the compartment and call it the number of molecules, we have to do at that part, so we have to do a random, right? So we don't have a proof of that. It's a good point. But actually, it does not create a bias. Yeah, it is a good question. We don't have, so this paper had no theorem, right? But
But it worked, what like.
yeah,
it is
so
the
it's a good question, whether this pseudo compartment method, right, is it, how accurate it is, right? So we did not have a theorem for that. Everything is sort of pretty ad hoc. As long as, but when it changed to stochastic part, we have a number and then we just do Gillespie algorithm.
But are you using an SPDE on the left or is there any noise on the left side?
This is all
But
stochastic.
on the left side though, because you could use an SPDE
There's
here.
there's no, it's a
It
P.
would almost seem like you would want to use an SPDE there so that both sides have noise, but you're modeling one
Oh, I
is
see.
like the large scale limit and the other one is in small.
I see,
So
but we
that
don't
you're-
have a meaning for stochastic PD for space-time white noise in.
Yeah, but if you're not
Two
proving
levels.
things, you can still do it computations, right?
Oh, you mean discretize it, just
Yeah.
just stochastic ODE, right?
Stochastic PDEs, you just add noise. I mean, you can still run them, right? You can just-
Good
you don't-
point,
you don't
okay,
have-
then
yeah.
we're looking at actually a system of interacting STE,
Yeah.
right? Yeah, this is one way to simulate stochastic PD.
But you'd almost feel like the noise on the right side would mess up the solution on the left side, right? If you're
Yes.
trying to couple or trying to match things at the boundary, you would think that if there's some large fluctuation up or something, that's gonna pull the solution on the left up or something like this.
In
And
in
because
the,
on the left, you're getting like the average behavior, but on the right, you're seeing all the fluctuations.
yes, the fluctuation mostly come from like near the leading edge, right? The patchy pattern also kind of near the leading edge. The inside, when the number of particles are big enough, it's more smooth. But it's a good question. So you're saying, what if we take into account of noise even here, right?
Yeah.
But basically you are looking at like boundary condition depending on the fluctuation on the right.
Yeah,
And you solve the deterministic thing.
Then you still have a randomness on the left-hand side still because it's boundary conditions.
Condition is
random.
random, yes.
So the left-hand side is still random in a sense.
Yeah, it's better than purely deterministic and
Yeah.
oblivious of the randomness on the right.
yeah, but we also want to optimize efficiency, right? So if you use PD, it's much faster. So, and this also will move, right? So it's on the left now, but maybe after some time, it would becomes a thing on the right. But yeah, right, that's great. There are many ways that this kind of simulation can possibly be improved and be quantified. And yeah, we'd love to discuss with you any any better way to deal with this? Might be look at a simple model to prove a theorem, like how this hybrid scheme quantify efficiency and also the accuracy. Accuracy is easy to quantify, but efficiency depends on the algorithm more delicately. So I guess some theorem quantify accuracy, but not efficiency. So, and how to optimize between the two. I don't know. It will be interesting.
Great. Any other questions? All right, well, thank you.